Never stop learning, for when we stop learning, we stop growing. This is Teacher Evelyn. I welcome you all to our online class. We are now on the second week of our fourth quarter. I'm always stirred up to have you here, and of course, excited about the lesson for today. And that is exactly what it is all about. I need you to stay present, or if not, then I would like you to go over with our previous discussion. And also, class, please check and update our learning path for the quizzes and the activities online. So again, thank you students for you chose to be here, and I'm so proud of it. So are you ready? Then join me. So today our topic is all about meiosis. So the phases of meiosis where the sex starts. So meiosis is a type of cell division used by multicellular organisms in the formation of reproductive cells, or what we call the gametes, such as the sperm cells and the egg cells, or pores, the spores. So meiosis is similar to mitosis in several ways. One's mitotic process is also divided into same four basic steps. So we have the prophase, metaphase, anaphase, and the telophase. So like mitosis, karyokinesis is followed by cytokinesis. However, meiosis is different from mitosis in some very important ways. Okay, let us first read our learning objectives for today. After going through this presentation, you are expected to describe the changes that occur in each stage of meiosis, make graphic organizer to differentiate spermatogenesis and oogenesis, and appreciate the significance of meiosis in reproduction, variation, and in maintaining the chromosomes number of species. So this is a short recap. Previously, we have discussed about the stages of cell cycle and how we produce body cells in a process we call mitosis. Let us have a short review by identifying the stages of mitosis in the following illustration. Okay, so here we go. Okay, so every species has a specific number of chromosomes. The chromosomal number is either diploid number, which represent the 2n, or the haploid number that represent the n. When you say diploid, means the nucleus of a cell contains two sets of homologous chromosomes. So class, please try to follow. The diploid number represents the 2n, or the haploid number that represents the n. So diploid means the nucleus of a cell contains two sets of homologous chromosomes, half of which are given by a female organism and the other half from the male organisms. Okay, I hope it is clear. So Homologous chromosomes are pair of chromosomes having the same appearance, carrying both the genes that control the same traits except for reproductive or the sex cells, which is the gametes. So the egg cell and the sperm cells. So gametes are haploid in number. So haploid cells contain chromosomes in single set. Thus, it is equal to the half of the diploid number. Look at the following illustration to better understand homologous chromosomes, haploid, and the diploid. Okay, for humans, have 46 chromosomes. 23 of these came from the father's sperm cell, while the other 23 from the mother's egg cell. During sexual reproduction, the haploid sperm cell represent the N is equal to 23 unites with a haploid egg cell to form a fertilized egg or a zygote, which is now the diploid. So, diploid represents the 2n that is equivalent to 46. That is why humans carry 46 chromosomes. Okay? So 23 of this came from the father and 23 of these chromosomes came from the mother. So the zygote will grow and develop into a multicellular adult by mitosis. So for sexual reproduction to happen, gametes, which is the egg and the sperm cell, so which are haploid, must always be available. So what makes it possible to produce haploid cells? Okay, so the answer for this is the meiosis. So now, meiosis is a type of cell division. Okay? Meiosis is a type of cell division that results in four daughter cells, each without half number of chromosomes of the parent cell, as in the production of gametes and plant spores. So meiosis, again, is the cell division that produces reproductive cells in sexually reproducing organisms. The nucleus divided into four nuclei, each containing half the chromosomes number, leading to gametes in animals and spores in plants. So meiosis reproduction division. Meiosis reduction division. Meiosis is divided into meiosis 1, this is the phases of meiosis, and meiosis 2, which are further divided into karyokinesis 1 and cytokinesis 1, and karyokinesis 2 and cytokinesis 2 respectively. So the preparatory steps that lead up to meiosis are identical in pattern and named to interphase of the mitotic cell cycle. So interphase is divided into three phases. Okay, so the major events and important characteristics of each stage during the interphase are summarized as follows. So, we have the growth 1 phase or the G1. In this very active phase, the cell synthesizes its vast array of proteins, including the enzymes and structural proteins it will need for the growth. So in G1, each of chromosomes consists of a single linear molecule of DNA. So now for the S phase or the synthesis phase, the genetic material is replicated. Each of the cell's chromosomes duplicates to become two identical sister chromatids attached at a centromere. So this replication does not change the ploidy of the cell since the centromere number remains the same. So the identical sister chromatids have not yet condensed into the densely packaged chromosomes visible with the light microscope. So this will take place during the prophase 1 and meiosis. Now, for the G2 phase or the growth 2 phase, this phase as seen before mitosis is not present in meiosis. Meiotic prophase corresponds most closely to the G2 phase in the mitotic cycle. So the G2 phase usually preparation for the cell division, assembly of proteins such as the microtubules, which are hollow protein fibers that cause the chromosomes to move during the cell division. Okay? Just like mitosis, meiosis starts with an interphase as well as the other stages of cell division as what we have been discussed in the previous module or in the previous presentation. But in meiotic division, the parent cell divides twice in which we call the meiosis 1 and meiosis 2. So each meiotic division or meiosis 1 and 2 have stages. So we have the prophase, the metaphase, the anaphase, and the telophase. So study this diagram in the next slides. Okay, before I will discuss the stages of meiosis, I would like to give emphasis how meiosis is different from mitosis. Okay, meiosis results in the production of daughter cells containing half the number of chromosomes of the parent cells. The resulting daughter cells with half the number of chromosomes is what you call a haploid cell that represents the n or the number. And the daughter cells that are produced after meiosis are not alike because of the manner the chromosomes divide. And the last, there are 4 daughter cells produced after one meiotic process because the cell divides twice in meiosis. Okay? So meiosis is divided into 2 successive cell division. Okay, so the first part, called the meiosis 1, reduces the number of chromosomes from diploid to haploid and number. So this stage of meiosis is also called the reductional division. So the second part, you have the meiosis 2, is very similar to mitosis. Okay? So again, both meiosis 1 and meiosis 2 are subdivided into 4 stages. Okay, now let's begin with the prophase 1. So what will happen in this prophase 1? Now, the chromosomes here starts to coil and shorten. Then the nuclear envelope disintegrates. Okay? So try to follow. It is also uh, described in this illustration. So try to follow me. The nuclear envelope tries to disintegrate. Now, homologous chromosomes pair by a process called the synapses. So you have here the homologous chromosomes. Okay? Then, exchange of genetic materials. This is where you can see now the genetic material is crossing over. So, may occur between homologous chromosomes when the chromatids are broken. So crossing over contributes to genetic variation. Okay? Now, for the metaphase 2. So metaphase 2, pairs of homologous chromosomes move to the equator of the cell. So the paired homologous chromosomes align, okay, as you can see, align the metaphase plate. The chromosomes and pair are now attached to the spindle fiber. Okay? Okay, for the anaphase 1. So what happened to the cell in the anaphase 1? Homologous chromosomes move to the opposite poles of the cell. So can you see how they move from pair from the equator? Okay. So now in anaphase 1, the chromosomes move toward opposite poles of the cell by spindle fibers. So the chromosome starts to separate, or they are now separated. So what happened to the telophase 1 or and the cytokinesis? Okay. So chromosomes gather at the poles of the cells. The cytoplasm now divides. Okay, class, remember, in telophase 1, in most organisms, the nuclear membrane forms. So this followed by cytokinesis. Okay? Now, let's move on to the meiosis 2. So for the prophase, what gonna happen now to the cells? Okay, the nuclear membrane disintegrates. So can you see a new spindle forms around the chromosomes? So for the metaphase 2, the chromosomes now align at the metaphase plate and are attached to their centromeres to the spindle fibers. So if you can see, chromosomes now line up at the equator. Okay? So for the anaphase 2, each chromosome is now divided into two sister chromatids. So again, centromeres divide, chromatids move to the opposite poles of the cells. So the chromatids now called the chromosomes move to opposite poles. Okay? Now, for the telophase 2 and cytokinesis, nuclear membrane is now formed around each set of chromosomes. Then the spindle fibers now disintegrate. Diba? Nawawala na sila. and the cell undergo cytokinesis. Okay, so nuclear envelopes form around each set of chromosomes, then the cytoplasms now divide. Okay, the major difference between mitosis and meiosis occurs in prophase 1 stage of meiosis 1. So if you notice that in meiosis, chromosomes pair with their homologous. The homologous chromosomes pair and entwine in the process we call synapsis. As the synapsis occurs, non-sister chromatids, so try to refer this in the next slides, overlaps and exchange genetic material, a process now we call a crossing over. It is the result to the change in genetic recombination. Our genetic material between parent cells and the daughter cells will not be strictly identical. So this event leads to variation, okay? Okay. In metaphase 1, homologous chromosomes at the equatorial plate then are undergo anaphase 1 and telophase 1. So take note that the number of chromosomes in each daughter cells after meiosis 1 is now reduced into half or haploid in number. Then each cell will now proceed to meiosis 2, passing once again through these stages: prophase 2, metaphase 2, anaphase 2, and the telophase 2. Since the cell now is in haploid number, unlike in meiosis 1, where the parent cell is a diploid. So each cell will now produce two haploid daughter cells, so that in all, there are four haploid daughter cells produced at the end of the entire process of meiosis. So I hope you can easily understood the difference between the meiosis and the mitosis. Okay, what is the importance of meiosis? All organisms have diploid number of chromosomes. Without meiosis, the chromosomal number will continue to increase. So hence, meiosis ensures that all the chromosomes number of organisms remains the same generation after generation. Meiosis also safeguards that each daughter cell receives a copy of each kind of chromosomes. Thus, each daughter cell would receive each kind of genes. The occurrence of crossing over and the independent assortment of chromosomes during fertilization ensure that the chromosomes are distributed to the daughter cells in different combinations, bringing about remarkable variation in the characteristics of the organism. So this variation in characteristics is very helpful for the organisms to adapt in their changing environment. So meiosis is the main event involved in the process of gamete formation. So gamete formation in male is called the spermatogenesis, while the gamete formation in female is what you call the oogenesis. So the gamete produced are the sperm cells in the spermatogenesis and the egg cells or the ova in the oogenesis. So both spermatogenesis and oogenesis involve the same steps in meiosis. So thus the resulting daughter cells are haploid cells and the two processes differ, however, in the number of gametes produced because four functional sperm cells are produced in spermatogenesis while only one functional ovum produced in oogenesis. Okay? So, meiosis and sexual gametogenesis is the process where sex cells undergo meiosis to form sperm cells. So, again, spermatogenesis in male and the egg cells oogenesis that is in female. So, please try to take note of that. So, spermatogenesis, again, the production of sperm by the process of meiosis. It begins at the age of puberty, 13 to 16 years of age, and continues until the old age. So, starting from a diploid to an spermatogonia, singular, and spermatogonium that will actively divide by mitosis so that there will be continuous supply of sperm all throughout the male's life. So this is how you are going to multiply your gametes or the male gametes. The male hormones stimulate the growth of spermatogonia. So one single spermatogonium will differentiate into one primary spermatocyte. At puberty, this one primary spermatocyte divides by means of meiosis, one to produce two haploid secondary spermatocytes. The secondary spermatocytes undergo meiosis too, and each cell produces two spermatids that are haploid. So this results to a total of four spermatids that will mature into form sperm cells. And the entire process of spermatogenesis results to four haploid sperm cells from one diploid spermatogonium. Oogenesis, the process of producing egg cells by means of meiosis. The production of oogonia by means of mitosis begins during the prenatal stage in the development of a human female. So the produced oogonia will be released in its lifetime. So again, meiosis 1 begins at nearing a birth of a human female. In meiosis 1, one oogonium transforms into one primary oocyte, which remain at this stage until the female is born and reaches puberty. As meiosis 1 finally complete, a primary oocyte divides into two haploid cells of unequal sizes. So the large cell is called the secondary oocyte and the small one is called the first polar body. Both cells enter meiosis 2 and the division process occurs again. So try to study the illustration of the stages of meiosis below and complete the table that follows. Okay. So the significance of meiosis. So it maintains the same chromosomes number in the sexually reproducing organisms. So from a diploid cell, haploid gametes are produced, which in turn fuse to form a diploid cell. It restricts the multiplication of chromosomes number and maintains the stability of the species. Okay. So maternal and paternal genes get exchanged during crossing over. It results in variation among the offspring. And all the four chromatids of a homologous pair of chromosomes segregate and go over separately to four different daughter cells. This leads to variation in the daughter cells genetically. Oh, we are now on the last slides of our presentation or discussion for today's lesson about meiosis or the stages of meiosis. So class, before I'll end, I would like to remind all of you to answer the quizzes and please do the activities, then submit it online. So I'm going to wait that. So maybe later or tomorrow I'll be checking your quizzes and the activities. Okay? So that would be all for today and have a great day. So see you next meeting.